to a rainy fall day of microbiology. So we're started talking about metabolism, and we thinking about this idea of respiration, went through the beginnings of that, and I think we had a homework assignment to look up what respiration is, okay, or at least get a different perspective by Googling it, wiking it, whatever the right verb is, I suppose. So what do people come up with? Or you just go like, oh yeah, I don't care, I'll figure it out, I'll wait till next time, I don't have to listen when you said. I don't care what you do, I hope somebody looked it up. Are you looking up now? Respiration. Anyone come up with anything? Nobody. The internet doesn't define respiration? Your book doesn't define it? Yeah, go ahead.
Yeah, which one?
Backwards Catman.
I had a form of aerobic catabolism in which an electron donor is oxidized with oxygen or an oxygen substitute as a terminal electron receptor.
Okay, we've been warned, or we've been talking about the oxygen part. Very good, thanks for that. Very good. So, oh, I just thought I was hearing something going, I want to say something. Okay, so we know about the oxygen. We wrote up the summary we usually learn in biology, general biology, and that's what they teach you since whenever you start learning about metabolism and oh yeah, everything's sugar, and sugar goes to oxygen. Okay, and CO2. All right. What's your name?
Connor.
Connor. Okay, Connor is excuse me, I'm still learning names, but I'll get there. Connor is saying that okay, there's these alternate electron acceptors. So look for something really electronegative, and when we start shifting between different oxidation states, like when we were talking about with sulfur, when you see sulfur with a lot of oxygen on it, or elemental sulfur, it's more oxidized, of course, than sulfide. So he's saying, okay, that could be a candidate. For nitrogen, which would be the more likely electron acceptor, nitrate, NO3 minus, or ammonia, NH3. Now, which one's going to be sitting at the end and waiting for those electrons to grab them up and get reduced? Which one looks more like oxygen? The one with the hydrogens on it, or the one with the hydrogens on it? Okay, how many people think nitrate, NO3, would be a good electron acceptor as opposed to ammonia? So we're going to start with how many people think nitrate? One. How many? So ammonia? Everyone else thinks ammonia? One. Two. Three. Rest of it, I don't know. Okay, wait. Let's see if your hands work. Okay, anyone, lift up your hands. Okay, guys, just a test check. I'm a scientist. I got to make sure the method I have is effective. Okay. Well, we're undecided. Look at so what do we know at this point? We know oxygen is accepting electrons and becoming reduced to CO2. We know that SO4 can accept electrons and get reduced to multiple different things. So now our question is, which is a better electron acceptor? Well, we'll just keep it simple. Ammonia or NO3. So the question is, which one? So, and it's okay to be, confused, or not know. That's why we're here to learn these things.
Can you use a different marker?
Yeah, they're,
Thank you.
didn't see it there. Do you need these again? Okay, oxygen. All these are kind
I have one in my bag. Do you want it?
yeah, eventually. So NH. I mean, I don't have to, you know what this is, but I'm just doing this so that you can put it in your notes. You know those chemicals, you all take in chemistry, so this is not new chemistry for anyone here. So which one would be the good, the more electronegative kind of compound? Nitrate or ammonia? Now let's vote. Nitrate? Okay, about a third. Ammonia? Okay, not committing? See, the non-committers, that's a funny part of you guys, because if I'm not gonna commit, how can I raise my hand? You know, so, we gotta think of a different method, maybe we can stomp our feet or something, I don't know. Well, it's nitrate, because, look at it, nitrate is, more like sulfate. Actually, I might have a better one of these. Okay, this is more, oxygen-like than this. This is very reduced. This is a great electron donor. Okay, just like in our way we learn respiration, how glucose is. Glucose is more reduced than CO2, so it has energy it can give to things. Okay, so that was one part of it. And Connor, what was the other part of the definition? You still have it handy?
No.
Okay. Well, I think I remember it. I thought you might have it right there, so we get the exact wording. But the other part was this electron transport system to move electrons. Okay, and we've already inferred that the electrons are going to somewhere, And in part 2 of the lecture, if we get to it today, we're going to learn about. I think you've seen this before, that the electrons move through this electron transport chain in the membrane, and they give off a little energy, a little more energy, and they're pumping protons out of the membrane. This should be somewhat, familiar to you. If not, we'll go through it again. Don't get too worried about it. But and at the end, they finally are sitting there, and I always think it's like the catcher's mitt. That little electron's like a baseball bouncing back and forth, losing energy, and now it's in pop. It ends up on oxygen, and it's able to grab a proton and start reducing oxygen to water. Okay. So the key is, in respiration, as Connor mentioned, you can have oxygen. That is respiration, because when we take the formula apart, that we have glucose plus oxygen equals CO2, water, and energy. When we take it apart, we know all that electron stuff is happening. That's a summary formula. That's what we learn, because when we start out in biology and energetics, we like to, kind of keep things simple, but then we start learning the mechanics behind things, because that's what we do as scientists. We like to know how that works. And we have Kelvin and a whole bunch of other people that sat down and figured out all these pathways. Okay. But that's aerobic respiration. If the electrons are bouncing down, if a organism has the capacity to have the electrons bounce through the electron transport chain and end up in nitrate, and eventually reduce that, they'll reduce down to nitrite, it'll reduce down to N2, NH3, maybe even to ammonium. There's a few other intermediates in there. Those of you who studied the nitrogen cycle in ecology probably know there's 5 to 7 different intermediates that you probably talk about. Then, that's anaerobic respiration. So what's different? What's the same? Well, the very fact that without, I know aerobic sounds like air, but think oxygen. Without oxygen, it's without oxygen. Does it still work as respiration? That's the part that's different. This is different than that in some of the bacteria. Sulfate, wherever it goes, is different than oxygen also. Okay. So, but the mechanisms, that's the same, so don't let that fool you. You got a different catcher's mitt, behind the plate, catching the electrons, but you have the similar process. Now, if you dig these things apart, some of the quinones and some of the intermediates in the electron transport chain are different, aren't all the same, but the same mechanism is there. You're bouncing electrons through the membrane in an orderly fashion, okay, and then with the same idea that you're pumping protons out, and now you're charging the membrane. You're making it into a battery, okay? The outside of the battery is positive, okay, the inside of the battery is negative, and that causes a chemoosmotic effect that when, at some point, by osmosis, by just plain physics, it's going to say, well, that's enough of this business. The protons are fighting to get back in. and you put a magic channel in the cell membrane, and that magic channel is called ATP synthase. And ATP synthase is sitting there going, okay, I got ADP, but I got no juice, I got no power. Oh, here comes a proton, bam! Captures a phosphate molecule, makes it into ATP, and we all know ATP is the energy currency of the cell, or whatever our teachers taught us. That's how I remember it. The energy currency of the cell. So that's the overview. Okay, so when you hear respiration, you think, well, somewhere in that cell, there's got to be a membrane, or if it's in your cell, the membrane's in the mitochondria. And then I got to look at who is catching the electrons at the end. If you don't like my catcher-mitt theory, come up with your own, that's fine. But when you look at the chain, that's just how my brain works. But if it's not oxygen at the end, and it's nitrate, it's anaerobic. Lots of things can do this. Iron can go in there and do it for some microbes. Sulfur, as we talked about, sulfur in different forms. Sometimes it starts out as sulfate, sometimes sulfur can be used. We talked about inclusion bodies. And I go, oh, I'm starting to get the idea here. You know, it's a relative thing. It's thermodynamics. It has to do with the oxidation state of the chemicals, and these microbes can use different forms of it. It's not always all about carbon. Okay, I get it. Now, we're talking about energy pathways. We're not talking about building, carbon molecules or tearing them apart. That's where energy goes into or comes back. Okay, that's our products. We're just trying to talk about how do we run the engine of this thing. Okay. So what I'm asking you to do is to kind of give up a little bit on that scheme you have in your head that you've developed since whenever you learn respiration, and give up a bias. Whenever you're respiration, think electron transport, think electronegative molecule getting electrons, and give up that idea that, oh, it's sugar and oxygen going to that. That is part of it. That's part of the story. This will then free you for the other thing we need to talk about, and that's fermentation. People I can't say always, many people, many of my students, and many faculty actually ask them sometimes, confuse fermentation and anaerobic respiration. Because the fermentation is actually independent of oxygen, but is generally an anoxic process. And we know fermentation, a lot of us, fermentation to some extent is used to make the gas bubbles in your bread. Okay, we like puffy bread, otherwise they'd be crackers, and I'm sick of crackers. Okay, or in yogurt, of course, famously alcohol. We get a bunch of sugar from different sources, grapes or malt, and we put in a yeast to do the fermentation. It goes anaerobic, it spits out CO2, it makes enough energy to keep things going, and it gives a byproduct. This byproduct that people enjoy is ethyl alcohol. In your muscles you get fermentation, and the byproduct is lactic acid. You're like, oh man, I shouldn't have. What was I telling you yesterday? He said, I had an easy day. Usually I run to Phoenix. Today I only had to run to St. Louis. You must have lactic acid build up. No, I'm used to it. So, these kinds of things are part of us. We get that. But that is fermentation. It is a low energy thing where people get confused as you start with CO2 when we learn it. And we say, okay, it ends up with pyruvate. Okay, now I got pyruvate, I got a choice. I either take pyruvate over to the citric acid cycle, goes around a few times, gives us NAD, a bunch of FADH, a bunch of reduction potential. Or it goes into these byproducts. So fermentation does not have electrons. Movement in the cytoplasmic membrane, there's electrons moving always. So I guess I've gone on a little bit on this, but I wanted to kind of get that precursor going in your head. So now when you look at this, we see diagrams that explain it. Maybe for a lot of us it's review, so sit back. For some of us, like, I never thought about that way. Good, wherever you're at. We're going to move through these lectures. Do you have any questions on that? That is a very important concept, to get into your head. one concept that will come up, and I will ask you a question on the exam on this. I will tell you, I have no problem. All the answers on the test, we're talking about, but I'm gonna ask you the difference between oxidative phosphorylation and substrate level phosphorylation. Now, right now, you might be like, oh, I've kind of heard that, okay? But, are you putting phosphorus onto ADP using an oxidation method, or are you just moving bonds around and thus shifting the substrate? Maybe moving bonds from double bonds to single bonds, and eventually moving some bonds so you can get a phosphorus off of a carbon and plop it onto an ATP. We see substrate level phosphorylation in glycolysis, which is common to both aerobic pathways, but it is the main pathway in fermentation. That is why fermentation isn't oxidative. It's moving phosphates around with substrates, with substrate alterations. So those are kind of overall themes. Okay, if you have them in your pocket, great. Sit back and relax. Let's see if we can get this going. Computer said, don't talk so much. Show more pictures. All right. So I like to start with definitions. We're scientists. It's good to know what we're talking about. We know a little bit more about respiration than we did coming in. Metabolism, the sum total of all the chemical reactions. Okay, that's what metabolism is. Some of those reactions are going to release energy, and those are catabolic. Okay, and if you're going to take, you need energy like to make things, to make your proteins, to make more DNA, to get the idea. Then it's anabolic. Okay, we're going to probably focus a little more on catabolic reactions here and leave anabolism for when you take probably biochemistry and things like that. But we'll kind of point at it as we move. Now, those of you in lab, we've already talked about nutrients, and we get mixed up what nutrients are. We think, well, I know they're in soil. If I have good nutrients, I can grow plants. I know I need nutrients. So let's just get it out in the open. Let's talk about it. Nutrients are basically the things that make up the cell. They contribute to metabolism, to the catalyst, to all the things. So the nutrients your body needs is in proportion to the composition of a cell. Now, on some level, that makes sense. We need more carbon, more hydrogen, more oxygen than we need things like phosphorus or trace elements like manganese or zinc, which we know we need, especially if we use alcohol dehydrogenase and things like that. So our macronutrients are here, and a lot of people like to make up little, what do they call them, mnemonics or whatever it is to remember it. So C. Hopkins Cafe, mighty good, nah, okay, that's a way to remember it, but what are they? Okay, here's our organic molecules plus oxygen, phosphorus, okay? These, without saying, the chalk part of it, well, let's just see if we know. We know all the macromolecules, we've been through them. We know they have CH and O. Which of the 4 macromolecules do we see phosphorus in? What are the 4 macromolecules? Okay, I know it's a rainy day, we're warming up, but let's kind of get a little conversation going. I've gotten you guys, I'm going to go with this guy, the white cap man. Tommy, okay, thank you. That helps me. I will get things. Connor,
lipids,
Tommy,
lipids, carbs, proteins, nucleic acids.
Phosphorus in.
I was just
the 4 ones, yeah. Thanks. I got so caught up in Tommy, I was thinking, it's like a song from The Who. I'm all mixed up. Okay, so proteins, nucleic acids.
Lipids and carbs.
Lipid and carbs, okay, or polysaccharides. Same idea, good for you. Now, in those, predominantly, where are we more likely to see phosphorus of the big 4? Go ahead, give me
Lipids.
lipids in the phospholipids. There you go. It's in the name. What else? And so lipids, proteins, maybe, but not a big deal. Nucleic acids. Yeah, the phosphorus has that dianhydride bond. It's the backbone and keeps everything together. Nucleic acids and ATP. We see this. Okay, phosphorus is easy. Where did I go? Okay, so Nitrogen, which of the big four can we definitely say nitrogen's a big player? You know, we can say, well, you know, I did see some nitrogen in those lipids, you know. There seems that there could be some, you know, attached to the phosphorus. Let's let that go. In general, yeah, maybe not. Okay, so what about, so that takes all the lipids for right now. Where do we know we see nitrogen? I mean, where is it a player? Nucleic acids have the nitrogen bases. Lots of nitrogen in there. And pick any base. Okay, good. We got that. And any others? Proteins. Sure. Amino acids. The amino group has it. In fact, and you know, amino, so let's just do a little calculation in our head. You know, we figure a cell, you know, if we get rid of the water, 50 to 70%, depends on what the is, we just have the dry weight, okay, we know that's about, you know, then 50 to 60% of the cell. What's the most predominant of the four macromolecules of the dry weight of the cell? Is this the dry weight of any particular cell mainly lipid? Is it mainly nucleic acid? Is it mainly protein? Or is it mainly carbohydrate? Pick one of the four. I do think carbohydrates, but you know, think about it. Carbohydrates are energy. They kind of move through. They're a little bit in the bacteria with the structure in the cell wall. Okay. But so carbohydrates is a close second.
Lipids?
Lipids, where are we seeing lipids mainly? It's cell membrane. Okay. So that it's a player, but lipids is not the answer for the most predominant. So we're down to 2. Well, we know nucleic acids. I mean, they're cool. All of it's necessary, but they're just not by weight a big deal. Proteins. Think about it. They're in the membrane. All the enzymes have to be proteins. Okay. The ribosomes all have protein, and on it goes. So they're structural, functional, and throughout the cell. So much so that when we think about the nitrogen of proteins, it's about 6.25%, about 6%. So 6% of the dry weight is that one element that makes that up. So you know, we start thinking, oh, so we need a lot of carbon, we need a lot of oxygen. Okay, anyone who grows or farms, you know, knows that we got to add phosphorus and nitrogen to make plants grow. It's the same for us. You got to be cognizant of the protein you're taking and so forth. And generally, we have nitrogen and phosphorus in a ratio of about that's needed for life about 10 to 1. You need 10 times more nitrogen than phosphorus. It works out that way. Talk to gardeners. Talk to ecologists. That's about how that works out. The famous Redfield ratio is in there, but it gives us an idea. Now, these other guys, potassium, yeah, it's around. It stabilizes membranes. It gets in there. It helps with some of the transport, calcium and iron. The same way, sulfur shows up in like methionine. Okay, cysteine, you know, the 2 of the 20 most common amino acids. Okay, magnesium and sodium, mainly we're seeing these in membrane transport and so forth. So we really direct our attention to the C hop and N and S are probably the main ones. So those are macronutrients. You need to eat those to build strong bodies 12 ways, or whatever the old commercial used to be. Micronutrients are still important. So anyway, what do we have here? We kind of talked about this passage. You can see some of the more details in here where some of these other things work. A lot of these salts are necessary. So if we're going to grow microorganisms, we need to put in a lot of carbon, hydrogen, and other things. And we do it. When you look at the media, we put in glucose. We put in magnesium sulfate. We put in salt. Salt has 2 things. Not only is it used for the cell, but it controls osmotic pressure. So cells don't either blow up or dry out, especially if they're clinical cells. Put it at point normal saline, about 0.9% or so. So, you know, there's some reasons for that. Now, you know, your Flintstone vitamins that you pop in the morning or whatever it is have a lot of your micronutrients. You know, what is all that stuff? What is it used for? Usually they're coenzymes. Okay, like I said, you need manganese and zinc and selenium, and there's a whole crazy list of them. If I'm growing microbes, or if you came to the lab and you wanted to, let's say we want to find some new microbes, okay, you have a good chance of doing that. You know, it's going to take you a while, but we can probably discover something new. Okay, well, I want a heterotroph, so I'm going to add all the carbon and stuff you just talked about. When we start adding the list of vitamins and minerals, we have to add trace minerals of like, you know, the list of like 20 to 30 compounds long, because we're hedging our bets. So does it have this enzyme? Does it have that? Well, it's unknown. We don't know how it gets its metabolism. So you'll see that trace mineral salts, trace vitamin salts, and yeah, I've had a project where I've wanted to culture novel microorganisms. It's one of the last phases of my research work, and I've had a few students start this, and they work through it very methodically, but it's very tedious, and a lot of people, you know, run out of time or interest. And I don't blame them. I say, well, you want to try it, we'll try it, but it burns people out because, you know, you're really trying to culture for the unknown, which is fascinating when you get it, but frustrating. And we have two species we're naming for Central Park, which we want to call them Nootropis central orbis, I think is Latin for Central Park. So we'll see if we ever get there. The Japanese are naming those guys right and left, or the Koreans, I think. We might not get a name in time. So we don't get to name a new organism. Anyway, so I'm on a tangent. All right, sorry guys, back to our class. So this is a nice summary table. We have seen this. Amongst the great discoveries in the world, which I rate to be the sewage system that the Romans came up with, the bicycle wheel, which I think changed time, life, and the way we live, is the periodic table. My goodness. You know, if someone said, you know, there's all this stuff in the world, but you know, it all adds up if you just change the protons and electrons, I think it's fascinating. But when we look at, we see some groupings. Our essential for all microorganisms are yellow, great. Essential cations and anions. Okay, we see, what is it, kind of a purplish color. There's a few of them, mainly in the kind of the area we usually think about with salts. Then we get into trace metals. Okay, trace metals for this aren't really used as electron donors or acceptors, as we're talking about. It's really used as cofactors. You'll see as you look through the book, I'll send for like nitrogen reductase. I'll say, oh, you gotta have manganese in there, you know, and you'll see that. And then we get into use for special functions. Okay, well that's kind of mysterious. And then unessential, but can be metabolized, weirder still, but kind of interesting to us. You know, we see things like mercury. You may see some other heavy metals. We find out that microbes can sometimes grab electrons from other things and make them less toxic. And people find that really interesting when we have heavy metal deposition, sometimes left by, let's say, fuel. A big problem has been fuel tanks that gas stations use leak. Now not so much, but you know. And with fuel there are metals and stuff, and now it's stuck in the ground, gets in the groundwater, people die and get cancer and stuff. And you find out, well, let's set some microbes down there. We know that these methanotrophs have an enzyme that can metabolize some of this stuff and make it less lethal. Let's let them do the work as opposed to, you know, digging up truckloads of dirt and putting it someplace else where it's going to leak. What do you do with it? You know, unessential, not metabolized. Iron feels badly here, but iron catches up. It's used in metabolism and some things. So, a summary. Do not sit and memorize it, but you know, as taking chemistry, this instantly makes some kind of sense to you. Kind of look around. That's my point of showing that here. We've talked about this. Nitrogen, oh gosh, I had it down by 6%. That must be for amino acid, 12%. Okay, phosphorus required for synthesis. Some of these other ones, then stabilizations we talked about, some enzyme activity, the membrane. iron showing up in the quinones and some of the iron proteins in the cytoplasmic membrane. So stuff we all need, but in proportion that the cell needs. Okay. There's a kind of a law, maybe you learned in cell biology, I don't know, but there's Liebig's law of the minimum, which states that growth of an existence of an organism is limited by the least found chemical, the least available chemical or element. All right. If you think about it, if you need everything to grow, you have carbon, hydrogen, phosphorus, but you don't have something for a crucial enzyme, iron, you can't grow. So in a way, it's obvious, but Liebig started to give us that idea of, well, for the opposite thing. We're trying to make bioreactors to make more ethanol out of corn. Sure, we got tons of corn. I mean, go outside, it's everywhere. We're shaking it out of our socks. We got corn every place. But to make it into ethanol, or to change from corn, which ethanol from corn is always debatable. Should we use switchgrass or something? Well, okay, we got plenty of stuff. How do we optimize the reaction? Maybe we need that minimum element, and that's a part of it. So we think about both proportion and necessity. There's nothing in here I want you to walk away or even memorize. I put this in here as is in your book because it's interesting to us, some of these different, I put it on here because I look at my vitamin bottle, I go, what is all this stuff for? Okay, and then this answers a lot of it. Oh, I need riboflavin for biotin. Okay, so that's kind of it. It's one of these, so I always wanted to know that kind of thing. So read through it, enjoy, get the idea though for that micronutrients are usually part, have that kind of function, enzymes, membranes, that kind of thing. Don't get too worried about, oh gosh, I gotta memorize what copper does. That won't be this class, maybe a different one, I don't know. So, and here's some vitamins, growth function, biotin. We usually take riboflavin. You always see that on back. I'm eating my Frosted Flakes. Oh, there's riboflavin in there as a little kid. That must be good for me. I'm happy they gave me riboflavin. So anyway, flavor proteins we'll see coming up a little bit, but we get to where some of these things go. So cool. Vitamin K. We'll talk about that. Produced by E. coli. Well, good for us. We're happy it does that because we need it. So I think I've talked on this just briefly. Who has a watch? Okay, can you say shut up at quarter after? Just say, Dr. McLean, please shut up. You do that for me?
Are you not, our class goes till 9:45. Oh, okay,
I need you to help me because I get off and I'm over here and sometimes I'm over there and I just need someone. I need an anchor. You're my anchor.
Okay, I'll be nice about it
Tell me to shut up. You won't be the first. But you just shut the hell up. Okay. Culture media to us is no longer really a mystery. It's still a mystery to us what's the right diet to feed my puppy dog. And it's still kind of a mystery to us now with the human microbiome. Well, should I really be eating this? Is there a probiotic that's going to take care of my metabolism or the way I think? The microbes are all in me. We don't know, but we have a pretty good idea. But if you look at our diet, when they give you the 4 basic food groups, I looked this up. Do you know where they derive that from? The government. And this isn't like, oh, it's a big plot or anything, but they had to recommend to people in the 40s, the 50s, in the war years, how to eat properly. They wanted people to be healthy. Do you know where the food pyramid came from? You know what I mean, the food pyramid? You eat more bread than nuts or something, and you eat, okay?
It actually isn't accurate, is it?
Huh?
It's not accurate.
Oh, no. I mean, it's accurate because it came from how you feed cows. they looked at that, well, what's the proportion when you're feeding cows, and I get a big cow and cut its throat and grab some hamburgers? Okay, so, it came from that, and, look, I mean, I looked it up and saw that. Look it up and say, Lemke, you're full of it. I found that it actually came from wherever it did. But I found it really interesting. So I was taught to eat optimally like, a calf. So it's like, oh, well, that's interesting to me. But now diets, you guys know so much more about that. If I'm going to feed a microbe, we're going to do the proportions of thing, a pure culture. I'm going to do a proportion of it when I just want to add different things, complex media. I might add beef extract. It's basically, they make that stuff, that's the broth after they take out most of the animal parts that they're selling, and then what they can't sell, they grind up in the hot dogs and sauce. You have no idea what's in there. And then the last stuff, they get beef extract from. But it's done the same way. Yeast extract done this. So when I'm making these proportions, like when you're making your stew or your soups at home, you're adding a cup of carrots. All right, do you know exactly what each carrot is made of? No, but if you add a cup, it's going to end up tasting kind of the way you think you'd like it to taste. So that is more like, so this is kind of the soup method. Defined media would be like, if you really need to control conditions, I want to find the microbe that degrades oil the best. Okay, I want to get rid of oil because oil is a pollutant, and I had a big oil spill. I want to get rid of it. I go down to the local parking lot and I scrape under some area where there's been oil dripping for 25 years. I think there's oil degraders there, and I put in as my carbon oil. I know it's a mixed carbon substrate, that's great, but I'm only going to vary oil and then some of its components. Oh, here's some aliphatics, here's some aromatics. Okay, everything else, I'm going to add the molar concentration of phosphorus, the molar concentration of nitrogen, control everything else, so I can find the guys that eat that oil the best. Okay, so define everything. I think we covered that. Selective media. We can put stuff in media that only certain microbes will grow with. If you're in lab, some of you were nice enough to help me out in culturing EMB agar. It selects for coliforms, because it has lactose and dyes that inhibit the gram-positive. Oh, okay, that selects for. So we can select based on metabolism. We can also have media that differentiates. The gram stain is a differential stain. It says, well, those guys are gram-negative and those guys are gram-positive. I can see the difference. In media, we have all kinds of tricks. We put in dyes that show up acid, or dyes that are picked up into the culture. So we can say with the EMB agar, coliform turns brick red. This is a brick red pure culture. That's likely E. coli. This one's pukey, pink, slimy. That's probably Enterobacter. I can tell the difference. Even though the agar is selected for coliforms, then it can differentiate between the two. So we use their metabolism. We use what we know to grow them, but we use what we know about them to tell the difference and find them. Okay, a lot of clinical microbiology is based on that. Even the new methods, the ELISA and stuff, are based on substrates and stuff. Here is kind of bringing full circle to the beginning of the idea that we start with energy classes and microorganisms. Okay, we're sitting here as the chemoorganotrophs. Okay, you might have heard yourself as chemoheterotrophs, a subsection of that yet. Okay, because there's also chemoautotrophs. Okay, what would be the difference between a heterotroph and an autotroph? Sometimes we say photosynthesis is photoautotrophic, or they're autotrophs. Anyone ever hear that? Yeah, okay, right.
They can make their own energy, right? And
Right.
the eat something to make their energy.
Well, but you always got to get your carbon from somewhere. So you got part of it. Okay, she's got the first part of the bullseye. Let's throw a few more darts at this thing. Get it done. Carbon source. We said some of them eat other carbon. Okay, that's what you guys do. You're heterotroph. I don't care what you eat, but you got to get carbon from somewhere. You just can't. So, but the other one makes their own, what did you say, makes their own energy. Autotrophs means that essentially, you're making, you're actually changing energy, you're transforming energy, but the source of carbon is CO2, or a single carbon cell, CO2. So in our chemoorganotrophs, this is fine for right now. Here's your organic chemical, okay, we're going to reduce oxygen, so we're going to oxidize this and get this and this. And there's some numbers you see are missing, but we get ATP. Yay? Okay, good. Here's some of these guys, okay, and you know, we can substitute the electron donors here. So we're, you know, some of the guys that use other things to collect electrons, there's other bacteria that can donate electrons from other sources, okay? We can see when we take the hydrogen off of hydrogen sulfide, okay, we can oxidize it to sulfate. See, they can make it go both ways. We can get some electron up there. Here's a whole bunch of electrons, okay? And just substitute here electrons or hydrogen, same thing. Okay, we're gonna reduce oxygen and we're gonna get ATP, okay? So I'm seeing here, I have a reduced compound, and I'm going to oxidize that to get energy, okay? What's the reduced compound over here that I'm going to take energy out of, and I'm going to eventually reduce oxygen? Glucose, good. It's just checking that you're following that, and then phototrophs are in there. It turns out that we have a few more types of metabolisms that we want to talk about. Get to know this one right now. Okay, if this is new to you, this is kind of stuff you didn't pick up probably in other classes too much. Let's talk about microbes. All right, yeah, enough of that. Let's move on. Okay. But yeah, so anyway, you know, STAR. I need to pay attention to these terms and what's going on there. Okay, we'll be coming back to that. So bioenergetics, here's some more definitions. I'm going to get through a few more of these before I have to stand back. Energy is defined as work, okay, chemical reactions. Some is always lost at heat. You know that. You have heat. Even the little microbes put out heat. They just don't control it as well as you do. You know, you have heat because your body's saying, oh, I got a bunch of stuff, but I also have cooling systems and stuff in there to keep it pretty steady state. When you let a microbe in earth and you let off heat, the earth doesn't really warm up. You know, it doesn't give off a lot of heat. Okay, we talk about stuff in free energy. Really here, I just reintroduced this to you so we have our terms in place. If you're losing energy, it's exergonic. If the reaction takes in energy, requires energy, it's endergonic. And what we need to do a lot of times in looking at our energetics is to calculate the free energy yield and the free energy of formation, GF, under standard conditions. Those of you who like this kind of thing, I'm happy to sit down and talk more about it. Go to the appendix, and all the different reactions that go on in the cells are listed there in their free energy of formation. We used to do a lot more work than we do now. If we ever will teach advanced micro, we'll do more of it there. But here we just, I think I have some simple examples. Some free energy of formation. Water's pretty high, okay, potentially. Glucose is kind of in the middle. Methanol, we don't think about methanol. Yeah. You are a good person. Okay. I'll finish up on the next slide. Thank you for that. So, you know, just kind of see, wow, this is a good battery for our cells to burn from, right? Okay. Methanol, we start learning that the one carbon cycle in nature is actually a very active and important, but it just really doesn't yield the amount of energy in there. I'll stop here. Look this over. I'm not going to ask, if I ask anything on this, and I think you've done this, I just brought this up because I remember doing this in freshman. You know how a reaction is, substrates give you products, you know, you then put the products, the energy of the products, you subtract the energy of the substrates, you go in and look it up, you realize oxygen is divalent, it doesn't really have anything. So for this one, to make methanol, you know, we put in 175, we looked it up from the table before, we subtract, we find out, oh, it will give you energy, that's cool. So look that over. I sometimes put something like this on an essay. I'll have like 10 essays, you pick 5 or something. So you know, something like this can show up on that. I don't dwell on it too much in our class. That's a good place to start next time. Okay. I know everyone says, oh, can I go home? Well, you can, but what we have here, I thought, I do a lot of discussions on Blackboard. I'll be putting some up, but I kind of thought it'd be fun to do this discussion in class and kind of break stuff up a little bit. So if you're here, you get to do a discussion. You can do it. In this discussion are word problems. I have 3 word problems. We have a half hour. I think we should be close in a half hour. If we stick to it, and don't talk about what happened on the reruns of Friends last night, we'll be okay. And what you can do is you can work in groups. I don't want the groups to be more than like 3 or 4. I don't want like the class to be a group, okay? Or if you're like, you know, I hate working in groups, work on your own, okay? What you're probably gonna need is access to a calculator, okay? Or like, do your phones have calculators, I thought? Is that okay? Did I say something wrong? Okay, so we're going to do some calculations. This is called so this is locomotion and scaling. You have a little bit of energetics in here, but I actually give you the energetics you need. But when we're thinking about movement, we just talked about it, and we've been talking about scale, what it means to be small. Okay, so what I'm going to do is I have 3 different slides. I thought what we could do is break up into our groups if we're going to have them. And then I give you the question, you can work on it a little bit or write down the essential parts of it. We go to the next one and go through these questions and then just work on them. And I can always show maybe there's a way I can make all the slides show up at one time. I think I can do that so you can just see them all at once. Okay. You follow what I'm talking about, or did I skip up there too far? So this is kind of something to break things up, to think about what it means to be microbial. Scaling means what it means to be small. So it's that kind of thing like, oh, the scale for the sperm to move from the place of ejaculation to fertilizing the egg. If the sperm was the size of a 6-foot person, it would be the equivalent of swimming from Vancouver to Anchorage, Alaska. And the amount of energy it would take would be, this is scaling up from things, okay? You probably learned that at some point. It's a real famous sperm thing. You know about sperm, right? Okay, so, I won't tell you about it. It's a big secret. You have to do with storks. Okay. So wait, let's get in our groups first. I mean, if you're going to just work by yourself, you're there. If you want to work with your buddies, kind of do a little dance or a cluster over to them and do that. Someone you want to work with is on the other side of the room. Great. If there's someone you've been looking at, like, I really want to meet that person, now's the time. I'd like to keep it around no more than like 4-ish. Aren't effective past that.
This is way too big.
Oh, you jumped. Hi. I don't want this to get stepped on. These are really expensive. Okay, just kind of do that. Tick-tock, tick-tock. Make sure your friend, if you don't have a calculator, buddy up a friend or a computer that has one. All right. So we got 4, 3-ish. Guys are great. And if you're doing it yourself, I'm the same way. It's like, I like working on my own. Okay. Longer it takes for us to do this, the less time you have to calculate. So what we got here, 1, 2, 3, 4, 5? You guys are always breaking the mold. Okay. Now, just so you know, now, take out a sheet of paper or tell someone you're the writer. Okay, on the top of that paper, write the people, listen to me now, because I don't want to talk again, and I'm not going to talk again. You guys want to keep talking? Less time for you. Okay, so, she's going to talk in. Okay, so get a piece of paper. If you're in a group, print the names of the people. You're all in a group, you're all going to get the same grade, right or wrong, okay? So, that's just the way we're going to do it. And do that now. So you got to be able to hand this in at the end of the period. Okay, not too big of a build-up. These aren't the big. Are we ready to go? So here's the first one. Okay, there are varying reports on how fast different bacteria can move. A way of expressing movement relevant to size is in body lengths per time. We've heard of that, right? For example, the cheetah in full stride can move from, some say 16, some say 25 lengths per second, which works out roughly to 90 miles per hour. We saw that on Discovery. I went and looked up the body lengths. One report indicates that some bacterial species can move 50 to 60 body lengths per second. Wow. Therefore, the bacterium is 3 micrometers in length and can move 50 body lengths per second. How far can it move in 1 hour? Assuming we're not doing run and tumble. It's just running. Okay, so I don't want you guys to get hung up on other details. So express your units first as centimeters and do that simple conversion into inches. You should have the internet, otherwise it's 2.54 centimeters per inch. Okay, work on that one a little bit, or do you want to see all the questions at once? Do you want me to read through all the questions? Okay, let's do the next one, and then I'll try to get all 3 of them up here so there's 5 questions, 3 slides. Okay, so that one's pretty cool, huh? You get to see how fast, if you were the size of a bacteria, how far could you go in an hour? Okay, if a 6-foot human can move as fast as the bacteria that we just talked about, this is just notes from the previous slide, this bacterium, 50 body lengths per second, how fast could she, the human bacteria, move in miles per hour? Okay. A little bit of conversion on some of that. Okay, this is the last one. This is energetics. So E. coli rotates its flagellum approximately 270 revolutions per second. Remember we talked about that last lecture? I didn't get into the numbers. Here it is. Let's say that a species is 5 micrometers long in length and can move 50 body lengths per second. Oh, we just said that. If it takes 1,000 protons to move through the MOT protein to make 1 revolution, how many protons does it take to move the distance of 1 millimeter? So that's a pretty straight multiplication. But now let's take it and figure it out when it comes to glucose, something we'll be talking about more on Tuesday. 1 molecule of glucose produces a bunch of NADHs, some FADH in the citric acid cycle. This results in an export of 112 protons if you work it out. Okay, about how many molecules of glucose are needed to move E. coli the distance described in question 4, the 1 millimeter distance. Okay, so the key thing here, this is kind of background. Okay, yeah, but it results in this. So that's your multiplication. Now, if you're working in a group, you can talk this over. You know, if you need some conversions, I can either give them to you, or you can get them on the web just as easily if you're connected. Do you get the questions? What I'd like you to do, if you have time, and you can work hard on 5 questions, I think you can do it. Some are very simple. After you get your answer, talk about your group. What do you guys think of it? Like, oh, that's what I expected, or, huh, that's kind of interesting. I find this interesting because, or like, wow, this is a massive waste of time. Don't ever try to do this again, Lemke. You know, I am insulted. I'm good at that. Okay, just tell me what you think. And I'll be around to field questions, but let's see if I can get these up for you. Oh. They're there. It's a little smaller, but you can see them, yeah? Okay, if you have to come up. try your best, okay? Even if you're to the point where, like, I don't know what I'm doing, I'm gonna give you partial credit. Let's make it 2 points per question. So, to answer the question, do some calculations, show your work. You know, if you went wrong someplace, that's okay. And then tell me what it means, so that'll be the other point. So you can always talk about what things mean. So, 2 points. If you're working by yourself, you can think about things mean. If you want to reposition your groups now, I better find old Jeff over there. He's a great mathematician. Go ahead. Go ahead. Okay? And start on the one that you like. Maybe the one that's confusing you. The next one, I don't care. Does not bother me. What order do you do it? Oh, divide count. Is it doable or is it an impossible question?
I think it's 52.
I'm reading this, but if you close, I'm gonna have to grind. Isn't that kind of? Okay. I mean, I don't want to get in your space or anything. I guess there could be many things. Okay. Get that? Now, if someone's watching, you can divide and conquer also. Someone will work on, so you guys are. So, at the time, you might want to say, well, why don't you guys work on subdividing? There's no law against that.
The only one I can't do, sadly, is number 2. I do not have the conversion of inches from meters memorized.
Oh, I just said 2.54 centimeters in an inch. And you can convert that if you need inches to centimeter. Yeah, but 2.54.
Thank you very much.
2.54 centimeters in an inch. I believe I'm correct on that. I've been using this, so I've been using for the last
1,000 proton.
That is, what is that?
There's like protein per revolution.
5,280 feet a mile, what else you need, but stuff like that. Would you like me to pose by that? You want me to pose with me? See how polite she was? No, that's okay.
Wait, yeah.
If you're working in a bigger group, it's okay to subdivide, give somebody 2 and 3, and you do 1.
Yeah, that's I mean, that's not bad.
Well, this, you're basically, if you're basically that, day 6 is basically say revolutions per second.
This one, scientific notation on this one, please.
I'm confused on how.
Oh,
Oh, well, the body length is, if I have something that is a body length this long, and I move 1, then I've moved 1 body length. So if I'm only 5 microns, you know, and I move 1 body length, I've moved 5 microns. So 50, or whether it's 3 or 5, let's just say it's 5 microns, and it's 50 body length, that's 250 micrometers.
The question is how many protons it takes.
When you do the body length, yeah, if your bacteria is the, yeah, oh yeah. Yeah, the 6 foot human, yes. Yeah, we're assuming a 6 foot human, you know, rather than keep it simple. Yeah, so now the bacteria all of a sudden is the size of, you know, like this guy, 6 feet tall. Now that it's moving 6 feet every time it's moving the body length, and that just scales it up. So if the bacteria is the size of you, how fast would it go, how far would it go? That's the idea, it's it's a fun skill. Are you having fun?
So for that one, we need to take.
We're having fun. What would make it more fun? What's that? Oh, who brings in the six way into this? Well, enjoy it what it is. At least think through it and work the parts of it. You think about the meaning of it too. So maybe you're working math, you're thinking about